Okay, it's 9 o'clock, so we're going to start with the Chi-squared test now. This is chapter 16, part 2b. So what is a Chi-squared test? It is a statistics test. So we're going to start off with some introductions to statistics. First of all, we have 4 statistic tests to learn in this particular A2 syllabus. The two here, t-test and chi-squared. And t-test and chi-squared is mainly to test for a significance of difference. What does that mean? This means you take 2 sets of data, and the test will tell you whether the 2 sets of data is different from each other or they are pretty much the same. Okay, we'll do an example later, and maybe you'll get the idea of significance of difference. Okay. And number 4 is to test for correlation. So, is this, if this increases, I'll say that again, right? Test for correlation means that, hey, if y increases, does x increase? If x increases, does y increase? And is it statistically significant? The correlation, and maybe how correlated is it? Is it strong correlation? It means if it increases, it surely will increase, or is it a weak correlation? If this increases a lot, this other thing only increases a little bit. Now again, I'm not going to go to detail yet. We will do that in the chapters to come. For now, we're going to focus on chi-squared tests. Okay? Now, I just want to know, you guys did stats already, right? You took the stats paper already, right? Right. For maths, yes, very good. Means this is not that hard to you, because honestly, statistics in bio is very easy. You just need to know certain words to write. Okay, right. So let's start off with some introduction. I think you already know this stuff, but I'm just gonna say it anyway, just so everyone is clear here, and just in case you don't take maths. Also say, I'll give back to Mr. Aoyong already. Okay, never mind. I'll start again. I'll assume you know nothing. Right. So types of data, very important for the statistics, because different types of data require different ways of interpreting its significance. Yeah. So we have qualitative data, and qualitative data means usually not numerical, so categorical. It could be like unordered categories, so nominal. So for example, male, female, smoker, non-smoker, these are categories. Or it could be ordinal. These are also categories, but instead of male, female, maybe you have, rate this out of 5. So 1, 2, 3, 4, 5. This is not really a quantitative per se, it's ordinal. We consider it qualitative in bio, okay? Quantitative, which is the second type of data, is to do with numbers. Okay, it could be continuous, or it could be discontinuous, or what we call discrete. Now, continuous means it can take any value within a range, i.e., you can have 0.001. For example, height in cm, you can have someone that is 150 cm. I'm 150 cm. I'm very short. Right, you can have 150.1 cm. You can have 150.11 cm. You have all these little values in between, right? Many decimal places. Whereas when we talk about discrete data, discontinuous data, is usually whole numbers. So number of visits to the dentist, number of bubbles, number of cells in the field of view. These are discrete values. So when we talk about discontinuous and continuous data, we will expect different kinds of distributions as well. With the continuous data, we usually expect a normal distribution. So if we were to take the heights of everyone in the room, okay, heights would be the easiest example here. We can plot it into onto a graph, okay, and we'll realize that our values tend to be in a null distribution. There will be a lot of people who are average in height. There'll be some that's super tall, like Fujin here, and there's some like super short, like maybe me and Ruwei, probably. Yeah. So I'm sorry, Ruwei. I hope you are not insecure about your height. Okay, never mind. Most people are average height, and we expect this distribution out of continuous data lah, usually. Okay, so for example, weight as well. Let's not talk about weight, sensitive one, more than height. But the second kind of distribution we talked about is discontinuous distribution. And this has to do with categories. So this could be qualitative data that are, or even discontinuous data that we are collecting here. And they're usually not distributed, and usually we express it in the form of bar charts. Now with different data, okay, it's very important to know what kind of data you're dealing with, because this influences what statistics tests can be used. And each stats test, as you know in maths, have different criteria. So there are a few assumptions of what's going on. Now, before we go into the assumptions for chi-square test, I want to bring your attention to something called the null hypothesis. I just want to know, you learned this in maths. You took statistics like in October, November, right? So that's not too far ago. The last time I took a statistics test was in uni, and maybe the harder one might be A-levels, which is like, I don't know how many years ago. Can someone tell me if you have learned the null hypothesis before? Anyone in the chat? Hello? Nope. Okay, thank you very much. So you've never heard of the null hypothesis before. Never mind. So far you have learned what is a hypothesis, right? Null hypothesis you can think of is opposite of a hypothesis. Null hypothesis we usually abbreviate it as H0. Okay, so null means zero, right? So H stands for hypothesis. So H0, null hypothesis, is all about what you want to disprove. So it's a statement to assume there is no significant difference between two sets of results of data. Usually, think about our usual hypothesis. When X increases, this is a hypothesis. This is H1, a normal hypothesis. When X increases, Y increases. Yay! Right? There is a correlation. Or when you say maybe X, or maybe a hypothesis could be X is bigger than Y significantly, for example. But a null hypothesis is terbalik from that, right? It's completely opposite of that. It's saying that, hey, there is no significant difference between two sets of results and data. I'll tell you the significance later, okay? We say in a null hypothesis that the differences present are not significant and due to chance. So for example, if we want to use the XY example again, we would say for this hypothesis, the null hypothesis, the inverse of that, you know what inverse means, right? The opposite of that is that there is no significant difference between X and Y. Now, in a statistics test, what we test for, remember just now we said, right? We test for in t-test and chi-squared especially, whether it is significantly different or not, whether X is significantly different from Y. After we do the test, we will need to reject or accept the null hypothesis. Okay, so for example, if your hypothesis, let's just choose one. Your hypothesis is, okay, X is bigger than Y. This is our example now. And this is our null hypothesis. In the middle of the experiment, there is no significant difference between X and Y. Imagine we do our chi-square test that tests for significance and difference. And the chi-square test that we will learn later tells us that, hey, our data is significantly different. Okay, this is maybe result of Chi-squared test. Chi is big letter X, by the way. Right, Chi-squared test. Maybe it's yes, there is significant difference. So what do you do with the hypothesis? Hey, I can accept my, I can reject my null hypothesis because it says that it's significantly different, right? So I can reject this. And I can say that this may be correct. Why may? Because the chi-square test tells you if it's significantly different. It doesn't tell you if it's more or less. So that's a function of the null hypothesis. It's something for you to reject or accept at the end of a statistics test. I think as we go along and do an example, you get the idea of it. But for now, just remember the formatting. This is the format. So you can get rid of all of this. Don't care first. Later we will figure it out and have a better idea of what's going on when we do an example. Just remember the formatting. There is no significant difference between this fella and this fella. And this data set A and data set B words can be substituted by anything. There's no significance between something and something, right? I know it's quite abstract for now, but it will all make sense later. So, with that, let's start with the chi-squared test. We know, okay, so far that we have 4 tests to learn, 4 statistics tests, one of which is chi-squared, which tests for, again, whether something is significant or not. Okay, specifically, if the observed results are significantly different from the expected. So with chi-squared, there's always observed, which we represent as O, and expected, which is E. What do you mean by observed and expected? Okay, this has a lot of applications in genetics. Okay, for example, if Mendel predicted the ratio of 9331, the offsprings are not going to be exactly 9331, right? Maybe it's like, I don't know the exact numbers. Maybe it's like some random numbers because he did so many tests. Let me go back a little bit. So chi-squared test is very what do you call, important for this, because for example you have this number. Oops. For example, you did the flower color experiment and you got this as your real test result. This is your observe. You observe this many organisms. But what do you expect? You expect something to tend to the 3:1 ratio. So your expected is not this. What is the 3:1 ratio here? So I don't have a calculator beside me, but I'm just gonna find the calculator that I downloaded. Where is it? This is fun. Right, so we have 705 plus 224. Right, we divide that by 4. So we expect maybe 232 on one side. Where is my calculator? Oh shucks, it disappeared. Sorry guys. I accidentally closed it, didn't I? So the observed again is 705 plus 224. Right, this is the total number, and to find the expected one, we just divide it by 4. Right, so it's 232. Oops. 697 to 232. 697 to 232. This is exactly 3:1. This is observed. This is expected. Yes, we can see that 3.15 to 1 is very close to 3 to 1. But how to know if this is very different or actually the same? 3.15 is quite far away from 3, right? Compared to 2.99. So is it statistically significant, the difference? Or is it statistically not different? So is it basically the same? So this is what the chi-square test helps us with. Again, it will all become more clear as we do an example later on. So let's move on with what the assumptions are here. So again, chi-square test is to show if observed results are significantly different from expected results. Okay, only can be used if it's a discrete or nominal data, right? So it's a discontinuous distribution that we're expecting here. This is not for data that has continuous distribution at all. So very discrete. So there is 1 plant, 2 plant, 3 plant. There's no 1.5 plant. There's no 1.1 plant, right? It's very discrete data. It's whole numbers only. Now, again, to test the results of number 1, breeding experiment. So this has to do with genetics. That's why this is in this chapter. Genetics and ratios. I don't know why I sang that. And number 2, it also can be used for ecological sampling. So let's say the hypothesis is that, hey, there is more number of mud skippers closer to the sea than further away from the sea, for example. So how is that 2 sets of data different? So you collect data of mud skippers near the sea and mud skippers a little bit further upshore, and you compare the data using the chi-square test to find out whether it's significant or not. Again, need examples. We'll get there. So yeah, that is the chi-square test. This is the formula being used. So chi-square is what? Observed minus expected. You square it, then you sum it all up for all your data. Backtrack a little bit. Observed minus expected minus really square, divide expected, then only sum the entire thing up. Okay, you must realize that. Now let me show you one example starting now for you to get a better idea of what's going on. With all statistics tests, I think everyone's a bit blur at the start and then it becomes clearer and clearer later on. Okay, so let's do the p-plant example again, and this one you would know already. We did this just now, same exact question. The idea is round, crinkled, or yellow-green. Round is dominant, yellow is dominant. And we expect a 9:3:1 ratio. I think I misread the question. Ratio. So it should be 9:3:1, right? Right. Okay, I do have that in the slides. Now, let's say you have a total amount of 144 P offspring, and the expected one would be this, and this. Now, if you don't believe me, you can do some verification by yourself. Okay, how do you do that? Right, so you take 144 divided by 16, because 16 is 9 plus 3 plus 3 plus 1, right? This is 9, right? So 9, so you see, this is the ratio lah, 9:3:1. So you can see that crinkled green is 1, right? Supposed to be 1 ratio. So if you have 144 offspring is 9, and times 3 is 27, so 27, and then if 9 times 9 is 81. So this is following the 9:3:1 ratio. They will always give you the total number of offspring or allow you to calculate it. So this is your expected. This is expected. Okay, this is what you want. So what happened is you did the experiment, okay, and the actual observed is not exactly 9:3:1, but it's very, very close. So this is your observed data. It doesn't follow exactly the 9:3:1 ratio, Okay, but we're going to start with writing the null hypothesis for experiment to start off the statistics test. Number one, what is the null hypothesis? There is no significant difference between this and this. And in this case, in chi-squared case, between the observed and expected results. You want to be more specific? Can. There is no significant difference between the observed ratio of different phenotypes and expected ratio of different phenotypes. There's no significant difference. You can say that. But of course, opt for the easier one in this case for chi-squared, yeah, the observed and expected results. So now that we have a null hypothesis to start off with, okay, we always start off with a null hypothesis. Now we can find whether there's significant difference between observed and expected. Then we can decide whether to accept this or reject this. So, because this is a genetics test and observed and expected is involved, we're going to do a chi-squared test. Okay, so this is a table that they usually would plot for you, which is yay! They even draw the data for you, just need to fill in the blank, right? So I just need you to try and calculate this really quickly. It's just very easy. Right, you find the difference. Okay, you can even, is that an error? It's an error. 25 minus 27 is 2, right? Yeah. Well, we need to calculate this again. I'm not sure about that now. Right, so 7 squared 749. Right. Something's wrong with that. Brain no work in the morning. Hey, it is correct. I should have doubted myself. So 2 squared is 4, 1 squared is 1, 6 squared is 36, and you divide it with the expected, so 49 over 81. Okay, now I need a calculator. Right, you can do this on your own. 49 over 81. Right, I'm lazy, so I'm just going to do all one shot. Can I? Maybe. 4 over 27. Please tell me if I did something wrong because my maths don't work when I teach bio somehow. 7 plus 36 divided by 9. I actually could type that, I don't know why. Alright, so did everyone get this? Or is my maths failing? Just double checking. Seems about right. So this chi-square test is faulty. It should be 4.790. Well, another mistake to correct before I. So I will repost the slides again. Renee asked, how many significant figures should we write for the last column? I think 3 significant figures is actually enough. So actually, okay, or 3 decimal places. 3 significant figures or 3 decimal places. I know that sort of contradicts. Yeah. Let's stick with 3 decimal places, like this. Because later you see the data table, it's gonna make a difference. Now in the exam, whoops, it crashed. It's not my fault. In the exam, you do need to fill all these columns up if they ask you to. So you can do that. I don't remember values. You can fill in the value. So this is the number I got. Can anyone tell me if they got the same number, please? Thank you very much. Just verifying answers. If no one said anything, I assume it's correct. Yay, good. Maths is working. Okay, so this is the 4.790 is the chi-squared value here. Obviously, something went wrong here. I just want to see if... Yeah, something definitely went wrong here. It's okay, but we know it's 4.790 now. Now we are going to use the chi-square table. So for every test, there's a table we look at. We are going to calculate. We are going to look for the critical value at p equals to 0.05. This is the standard that is set by the, I don't know, scientific community of some kind. 0.05 means there is a 5% chance that is due to chance. What does that mean? It means a 5% chance, a very, very small percentage, very low probability that it is random. Which means it's statistically significant. Significantly different. This is what we mean. That's why we use 0.05. Critical value is a value in the table. Okay, later we'll look at it. All right, we're going to look for a critical value at this, and then we are also going to look for the degree of freedom. In this case, the degree of freedom is number of classes minus 1. So 9331, okay, for 9331, this means there are 4 different classes. Classes means categories. So degree of freedom will be 3 because it's 4 minus 1. Okay, if V confuses you, you can also write DF, I think. And then we will check if the chi-square value that we calculated, which was what again? Oh my gosh, it's 4 point something. Scroll back a little while. 4.790. You know what? I'm going to write it in chat because I will forget. Sorry guys. Typo problems. And then we're going to see what happens then. Okay, so let's look at our chi-squared table. This is our chi-squared table. This is how it will look like. Let's look at, again, p equals to 0.05. So this is a probability. P means probability. We'll look at 0.05. This means that there's, again, a 5% probability that it is due to chance, which means a very low probability that's due to chance, okay? And also, we realize that our degree of freedom is 3. So this is our critical value right here. The value in the table is 7.81. Now, what is our current chi-square value? Our current chi-square value is 4.790, which is smaller than this. Okay, if you will plot, can realize that this is small to big number, right? If we plot it on the table, it's between these two numbers, right? It's 4.790 right here. That number is wrong. So let's just use 4.790 here, which is smaller than the critical value in the table. Now, if it's smaller, means there is no significant difference between the observed and expected. Okay, why? That's because you need to be bigger than this critical value to have less than 5% probability that is due to chance. You want that this probability to be as low as possible. The lower, the better, right? To be sick. Why? Because if it's a very, very low probability that is due to chance, that means there's a very high probability that it is significant. You get the drift? You get the drift, yos? Okay, you know what I'm talking about this. So in this case, our value is smaller, and therefore there's no significant difference. Now, this is not bad news for us. Honestly, there is not bad news for us. Why? Because our conclusions are interesting, are good. Okay, let's look at the second part here, not the first part. What? So this is how we express our answers. Okay, just now I did say in words, let's do it again. So since our chi-square test value is lower than the critical value in the table, this means there's no significant difference result, which is a good thing for us because we wanted our results to follow 9:3:1 ratio. Now that we got our observed, which is slightly different from expected, right? We did a statistic test, but now we realize that it's not significant difference. Means that observed is about the same as expected. So we accept the null hypothesis. Okay, null hypothesis is what? There's no difference between observed and expected. This is true, right? So we accept it and we say that, hey, any differences is due to random error or chance. This means that our observed, right, follows our expected ratio. So they both are around 9:3:1 and there is no significant difference. So it's good. Our experiment has worked in this case. Okay. All right. What if our experiment didn't work? What if our chi-square value is higher than value in the table, higher than the critical value? Now this would be completely opposite to what the explanation we just said. So if it's higher, then there is significant difference. Then we have to reject the null hypothesis. Again, the null hypothesis is that there is no significant difference between the observed and expected. So when we reject it, we are saying that yes, it is significantly different. The differences are not due to random error and chance. And in the context of this experiment, which is our breeding experiment, right, our 9:3:1 thing, right, this is not desirable. We don't want there to be significant difference between observed and expected anyway. So it's fine. Okay. So using our example, this is how we would write it in exam. Okay. 4.790. Well, I did my slides. Right. So using our example, chi-square value is 4.790. Critical value at P is 0.05 equals at 0.05 is 7.81, right? Meaning that our chi-square value is less than our critical value. What is this? Go back. Okay, it's higher than, it's lower than our critical value, which is 7.81. These are the words that they expect you to use when they ask you to explain your result. So the probability of the difference occurring by chance is more than 0.05. You get it? So the probability of it occurring by chance, okay, so the probability of it being a random thing is very high, is higher than 5%. Statistically, we consider this high. Okay, so the null hypothesis is accepted. There is definitely, repeating the null hypothesis, no significant difference between the observed and the expected results. Means same lah. Okay. Do you guys need time to process? Or we can do another question. Any questions so far? Is it confusing? I hope it's not. Only the value here is wrong. But other than that, it's alright, right? Questions? Okay, no questions. Maybe one more example that you can do. Alright, so we are doing this again. But now we're not doing the 9:3:1 again. We are going to do this. We did this question just now. We draw the genetic diagram some more. We already draw the genetic diagram, so you don't need to draw again. Okay? No need to draw again. It's fine. We figured out that the ratio here, let me go to OneNote. Here is the next page. Okay, I should have written it here. Ayo. Okay, now I have to change all these stuff too. Never mind, never mind. Sorry, hang on. So go back to just now, right? We did this already. There. We did this today some more. So we did this. We figured out is 1111 already. We already drew the diagram. So go back. So the expected outcomes with its ratio, we already drew is 1111. Great, great, great. Next. So they tell you the total number of offspring is 124. This is the numbers actually observed. Can you please write a null hypothesis and calculate the chi-square value? We will look at the table together later, but can you write a null hypothesis and find the chi-square value now, please? Realize that the expected ratio is 1, 1, 1, 1, just like your previous genetic diagram. The Chisquared table will be given an exam, don't worry. You know, they ask you to hafal, man. No. Calm down, calm down. 1. Okay? I need my calculator. Why did I bring it back from college? Fine, I'll use this calculator. Okay, I gotta get a paper and a pen too. Man, I haven't used my pen for so long, it started working. It started drying up. Okay, when you're done, let me know. Sorry, yeah. Okay. I don't trust my maths anymore. I gotta recalculate everything. Just verified my answers. Should be correct now. I also got 4.452. Okay, so I think some of you already got there. So what you need to do is to find the expected. So this is observe. Make sure you find the expected. What's the expected? 124 divided by 4, which is 31. Okay, so it's 1111, right? So the expected is 31, 31, 31, 31. I didn't give you a table here, so it doesn't matter here. Alright, make sure you find the differences first before you square them and divide them. I'm not going to write the working. Follow the formula. Hey, you guys didn't ask for the formula. You guys so smart, huh? You guys remember the formula already? So chi-squared is basically, I don't write it right. Chi-squared is basically O minus E squared over E, and the whole thing is summed up together. And you should get, for the chi-square value here, 4.452, if you do all the working right. Okay, in the exam, if they ask you, no, they will draw you a table and you will have to fill it out. Okay, they are very nice. Honestly, they draw everything for you. So let's look at the null hypothesis here and answer the question. This is the chi-square value grid. Null hypothesis is, according to format, there is no significant difference between. Again, opt for the easiest one when it comes to this. Observe and expected results. Yay. Always it starts with there is no significant difference between this and that. Okay, so with the value of chi-square of 4.452, let's look at the chi-square table here. So as usual, we will always look at the 0.05 level of significance. This is what we always look at. Okay, degree of freedom is 4 minus 1. I'm going to write it nicely. 4 minus 1, and therefore is 3, so we will look at 3. And the value, critical value, is the same as just now, 7.81. Now, is 4.452 bigger? No, it is not, right? Our chi-square value, I'm just going to write here again, is 4.452, which is smaller than our critical value. So is it significant? Is the difference significant? The answer is no, it is not significant. Yes, we always look at the 5%. Okay, we can look at the smaller one, but we can't look at the bigger probability. Always look at 5% first, and then enter below. Okay, I will show you an example later on. So how should our examples be? How should our phrasing be? Okay, the first one should be, this first sentence should be chi-square value is which is smaller than your not our the critical value. Okay, at p equals to 0.05. Okay, so there is a more than 5% chance that differences are due to chance. So does this mean you accept or reject the null hypothesis? You accept the null hypothesis. And you say that, hey, there is no significant difference between O and E results. Okay, questions? I see it. I just had to finish my sentences first. Miss, what if the ratio of each classes is not 1 to 1 and the expected result for each class is different, then each E should we take? So it depends on your ratio. Okay, so if it's 9 to 1, then you divide the total number into a 9 to 1 ratio as well. If it's another different ratio, then you again divide the total number into that ratio as well. So it depends, the expected result depends on the ratio. Okay, you may ask, so we always look at the 5% only? Well, this is the cutoff point. Okay, this is the cutoff point where we determine something is statistically significant or statistically not significant. But if the cutoff point, if it's lower than this value, it's okay. We can say that, oh, it's significant at the 0.01 level. Can, because it's lower than 0.5. Means it's an even stricter cutoff point. The one bigger or cannot. But typically, classical examples, we all look at 5% only. Okay. For beginners, look at 5% only. Was now asked, why is it 4 minus 1? Well, there are 4 different classes, so 4 different phenotypes, right? 4 different categories. So it's 4 different categories minus 1. That is the degree of freedom. If you're asking the other question, which is, why is it not just 4? Why you want to minus 1? That one I cannot answer you because it's a statistics thing and a complicated maths proof behind. There is a proof. I don't understand it. It's on Wikipedia. I tried. It's confusing to me. I'm not a maths person. Okay. Someone asked also, must it be same or bigger than the critical value to be significant? Yes. You need to be higher than critical value to be significant. We'll do one more thing, one more example, and this one is an ecology example. Okay. One more question for a friend. So is it always number of categories minus 1? Only for chi-square test. For chi-square test, it's always number of categories minus 1. For other tests, it might be different. And I'll explain why later on. But for chi-square table, always number of classes, number of categories minus 1. Okay. All right. This number 3 is a past year from I don't know when, honestly. I forgot. I recorded it somewhere lah. Okay, and this is an ecological sample, and you can talk, and this is about moths. Now, the null hypothesis here cannot be just observed and expected. It has to be based on the context given, because this is no longer just straightforward breeding examples, not just genetics now. I'm going to do it with you, but I want you to process first and like calculate for yourself first, and I'll discuss this in short while we will look at the probability table together later on and figure out the mark scheme later on as well. Okay, so I'm just gonna let you stare at this for a while and we'll complete it together in a while. I found out which year it is. It's very old. But statistics hasn't changed in statistics test didn't change in CIE syllabus. So I'm just going to give you a few minutes to process because I do still want to do this together with you because you're new at this and I will spam you with past year later on. So I am going to start now. So what it's asking is, grassland grazed by goats, okay, was compared to grassland ungrazed by varying amounts of times. Moths were trapped. So it's not about the goats, it's about the moths. They have the numbers counted and the moths were released, okay. Effect on grazing on a species of rare moth or domesticated. So they want to see whether there's a difference between moth on the grassland graze or the grassland ungrazed. So the answer for null hypothesis should be there is no significant difference between numbers of moths in grasslands grazed by goats. Obviously, I'm like with a lot of past years, I follow back the question. Okay, grasslands grazed by goats or grassland ungrazed. And ungrazed grasslands. There's no significant difference between number of moths in grassland grazed environment and in ungrazed grasslands. I'm going to go back a little bit so I can fix that. Okay, so this is your data set A and this is your data set B. Alright, so this and this should not have any difference according to your null hypothesis. Okay, now let's look at the observed and expected results here in the Chisquared table. Now you realize here that hey, it doesn't have a ratio. So what shall expected result be? Now our expected result should be all the same. So we assume, okay, that they're all the same. All right, so how much is that? Bringing my calculator here. They don't tell us the total, so you have to find the total yourself. So 111 plus 90 plus 36 divided by 3 equals, it should be 80 expected for each. Okay, why? Because you want to find significant difference. You want to find if there is an effect. So you assume that then it has no effect first, and if these two are significant different, means there is an effect. Get it? Alright. This is assuming no effect. This one may show some effect. So if they're significantly different, you know that there is effect. So how do you do that? How do you find the stuff? You just do the thing. Can I do this by hand? Okay, yes. 44 times 44 is what? How to square on the keyboard? Doesn't work. Ah! Right, I'll just press it instead. 1936. Okay, you can do this on your computer as well. I think you'll be faster than me because I don't have a physical calculator. I left mine in college. Genius. Right, 181. Okay, then we have 11. No! What did I do? Should be 10. Crazy man. 100. What did I do just now? Alright lah. 44 square, right? Yeah. And then 114 minus 80 square. Oh, we have 6 more minutes left. Don't worry, we'll finish on time. 1156. And this is past year. So see, they do give you the table. The bad part is you cannot key in all at once. You have to calculate one by one like I'm doing here. Usually the number all very pretty. Oh no. I really hate this now. You can find 100 divided by 80 is 1.25, 1.25, 1.25, and 14.45. 1.25 and 14.45. Sum it up together and you should get 39.9. Is my maths faulty? You get the same? Tell me if it is. Anyways, you should get that 49.9. And we can look at the probability table now. Yay! So again, we will look at 0.05 level of probability. How many number of classes are there? There are 3 different categories here. So degree of freedom is 3 minus 1, which equals 2. So this is your degree of freedom. Number 2. This is your probability. This is your critical value. So your chi-squared is really big. It is 39.9, which means it is larger than the critical value. Which means that your data, okay, the percentage probability that is due to chance differences are due to chance is kind of low. It's definitely not due to chance. Something is affected it. How low is it though? Okay, so 39.9 is extremely big. So this is 5.99 as a critical value. 39.9 is all the way over here, right, on the scale. See, this is 5.999. It's bigger than this number here. So we can say that probability that difference of chance is due to, probability that differences are due to chance is, in fact, less than, okay, less than 0.1%. This is 0.1%. Means that it's so tiny chance that it is due to chance, which means definitely, almost definitely, 99.9%, it is definitely something else going on. It is very significantly different. The observed and expected values are very significantly different from each other. The null hypothesis is rejected. There is significant difference between observed and expected. Right? So you can see here what the other questions are. Okay, this is the last slide for today. So number of degrees of freedom, this is 2. 3 minus 1. Probability of the value calculated for chi-square. Probability of the value calculated by chi-square. So less than p equals 0.01. I'm going to check my answers. Do I want to teach you anything wrong? Yeah, definitely. Right. Or you can just, oh, did I miss 1 0? Yes. 0.001. Or you can just say P is less than 0.001. Okay, I think this is more straightforward. The next is explaining the significant result and suggest the consequences to this morph. Okay, and I'm just going to paste the mark scheme here onto the OneNote so we can see it together. the first thing we should talk about is how the value, okay, explaining the significance, right? So it is significant. Yay, it is significant. I'm going to use a highlighter instead. It is significant. It is not due to chance. You reject the null hypothesis. All this is 1 mark. Sometimes these 3 lines can be 3 marks, depending on the question. This one, it has 2 questions in one, right? So this is only just 1 mark. Suggest the consequences of the morph if grazing land is increased. So what it's saying is that, hey, look, this means that if it's grazed, there is much lesser morphs. So what is the consequence? Okay, maybe it's because, say, suggest ma, so suggest law, right? Maybe it's causing the population of moth to decrease. Maybe it's because the plants are eaten by the goats already, so there is habitat destruction for the moths. And maybe the increase in predators because of the grazing. Maybe less grass means fewer places to lay egg or hibernate. And maybe if grazing increases, is even more likely to become extinct. So this is answering the question. The second question about suggesting what would happen if the species of moth, the grazing land is increased. So yeah, that is an example of passive questions. I'm going to spam you with more by the end of this week. Actually, you know what? I'm going to assign it today. Yeah, I'm going to assign you 1 today, and I'm going to give you a week to do it. I will discuss it next week when we meet again. Any questions so far? If not, we are pretty much done. No? Alright, you can bring this and go and digest what's going on later on. And of course, I'm going to give you the exam, the practice so that you can go and figure it out as well. Okay, with that, class is done. See you tomorrow. Okay, we have class tomorrow. By the way, it's supposed to be Monday, right? But we moved to Thursday, 1 to 3 o'clock. So I'll see you tomorrow, guys. Bye.